Welcome to the lecture called Protein Synthesis. So in this lecture we are about to see the protein synthesis, how the process are beginning, the transcription, genetic code, tRNA, ribosomes, translation process and the protein synthesis process. So in the central dogma of life, how the protein is synthesized. The DNA is transcribed into mRNA and the mRNA is translated into the proteins. So this is the central dogma of life. In the first step, the transcription, it is the leading step to the gene expression. So this stretch of DNA is transcribed into an mRNA molecule, which is called as the transcription unit, and it encodes at least one gene. If the gene is transcribed encodes for a protein, the result of transcription is the messenger RNA, which will be then used for the protein process. Alternatively, the transcribed gene is encoded for the either rRNA, that is ribosomal RNA, or tRNA or other complements or the components of the protein assembly in the ribosomes. This DNA transcription unit encoding for a protein contains not only the sequence that will eventually be directly translated into the protein, but also the regulatory sequences that direct and regulate the synthesis of that protein. As in the DNA replication, it reads from 3' to 5' during transcription. Meanwhile, the complementary RNA is created from 5' to 3' in direction. So only one of two strands, that is DNA strands called template strand, is used for this transcription process. Other DNA strand is called the coding strand because its sequence is the same as the newly created RNA transcript. So this transcription is divided into three steps. First is initiation, elongation and termination. So in the initiation process, especially in bacteria, transcription begins with the binding of RNA polymerase to the promoter in the DNA. So this polymerase is a core enzyme that consists of 5 subunits called 2 alpha subunit, 1 beta subunit, 1 beta dash subunit, 1 omega subunit. At the start of initiation, the core enzyme is associated with the sigma factor that aids in finding the appropriate 35 to 10 base pair downstream of the promoter sequences. During elongation, one strand of DNA, that is template strand or non-coding strand, is used as the template for RNA synthesis. As transcription proceeds, the RNA polymerase traverses the template strand and uses the base pairing complementary with the DNA template to create RNA copy. So although the RNA polymerase traverses the template strand from 3' to 5', the coding strand newly formed RNA can also be used as a reference points, so the transcription can be described as occurring 5' to 3'. So this produces an RNA molecule from 5' to 3', an exact copy of the coding strand, and the nucleotides are composed of 5 carbon sugar called ribose, where DNA has deoxyribose in its sugar phosphate backbone. Unlike DNA replication, the mRNA transcription can involve multiple RNA polymerases in a single DNA template and multiple rounds of transcriptions. That is, amplification of the particular mRNA is happening. So many mRNA molecules can be rapidly produced from a single copy of a gene. So this elongations is also involves a proofreading mechanism that can replace incorrectly incorporated bases. The termination process is the final step where the two different strategies for transcription termination, which is RHO independent and RHO dependent factor, which is called as a protein, and it depend on the RHO protein or RHO independent protein process. Thank you.